Today, we're diving right into a seriously fascinating question. How exactly do scientists track, measure, and understand things they literally can't see? We're going to demystify one of the most powerful, brilliant analytical techniques out there and see how we can use light to uncover the hidden microscopic world of nanotechnology. So, let's start with the core problem here. Nanoparticles are, by definition, unimaginably small. They are completely invisible to the naked eye and, honestly, even to most traditional microscopes. If you're in the lab trying to build or study these tiny structures, how do you actually know what you've created? How do you check its size, its shape, or if it's even stable? You obviously can't just pull out a tiny ruler. We need a highly specialized tool to bridge the gap, a way to translate the invisible physical properties of these particles into data we can actually read. Here's our roadmap for today. First, what is UV-Vis spectroscopy? Second, how the spectrophotometer actually works. Third, we'll break down Surface Plasmon Resonance. Fourth, 4 key nanotech applications. And finally, number 5, why this is the ultimate nanotech tool. All right, section 1. What is UV-Vis spectroscopy? At its core, this is a fast, entirely non-destructive technique that measures how light interacts with a given sample. Specifically, it tracks the absorption or transmission of light in the ultraviolet and visible spectrum. So we're talking about wavelengths ranging right between 200 and 800 nanometers. By firing this light at a sample and simply measuring what makes it through versus what gets absorbed, we can uncover an absolute wealth of information about the unique optical and electronic properties hiding inside those materials. Moving to section 2, how the spectrophotometer works. So how does this machine pull it off? Well, it starts with a light source, usually a deuterium or tungsten lamp, that throws out a broad spectrum of light. This light passes through an entrance slit and hits what's called a monochromator, which is often just a prism or a grating. Now, think of this monochromator like a really strict toll booth. It separates that broad light into specific individual wavelengths, and it only lets one very specific wavelength pass through at a time. That chosen beam of light then travels right through the sample cuvette. That's a small, clear tube holding our nanoparticle solution. Finally, whatever light actually makes it through the sample hits the detector, and that detector measures the exact absorbance or transmittance. But to make sense of this, we need the math, right? Enter the Beer-Lambert law. It is an incredibly elegant and fundamental physics equation. A stands for absorbance. Epsilon is the molar absorptivity, which is basically a constant for whatever specific material you're using. L is the path length, meaning how wide that cuvette is, and C is the concentration of the sample. So the absolute game changer here is this. Assuming the path length and the material's properties stay the same, the amount of light absorbed is directly proportional to the concentration of your invisible nanoparticles. Absorbance goes up, concentration has gone up. It is a perfect mathematical bridge. Section 3. Surface Plasmon Resonance explained. To really grasp why this technique is so powerful, especially for noble metals like gold and silver, we have to talk about Surface Plasmon Resonance, or SPR. This is the secret sauce. Imagine the surface of a gold nanoparticle is completely covered by a cloud of free-flowing conduction electrons. When light of a specific wavelength hits that particle, that entire electron cloud begins to move and vibrate together, perfectly in sync with the light wave. Think of it like a massive crowd doing the wave at a sports stadium, everyone moving together. When this resonance happens, the nanoparticle absorbs a massive amount of that specific light energy. Because of that SPR effect, noble metal nanoparticles don't just give us a flat line of absorbance across the wavelengths. Instead, we get this massive, distinct mountain peak on the graph. We call this the SPR band, and its absolute highest point is known as lambda max. For gold and silver, this sharp peak occurs right in the middle of the visible spectrum, typically somewhere between 400 and 600 nanometers. And this strong absorption of visible light is exactly why a solution of gold nanoparticles doesn't look golden at all to us. It actually looks vividly red or purple. Part 4. 4 key nanotech applications. Application number 1 is monitoring synthesis. It's kind of like watching a nanoparticle being born in real time. When you mix chemicals to reduce metal ions into solid nanoparticles, you can put a sample right into the spectrophotometer and track it continuously. Over time, as more and more nanoparticles form, that SPR band literally grows taller on the graph. You can even watch the visual color shift, going from clear to a faint tint all the way to a deep purple or red as the reaction finishes. By tracking this, scientists can instantly nail down the optimal reaction time and know the exact second their synthesis is complete. Application 2. Determining size and shape. Let's talk size first. As these gold nanoparticles grow larger, say going from 10 nanometers to 30 and then up to 60 nanometers, their electron clouds shift, which completely changes how they interact with the light. On our graph, we see this as a shift in that lambda max wavelength. Specifically, as the particles get bigger, the peak physically slides to the right, toward higher wavelengths. We call this a redshift. So without ever needing to look through a complicated microscope, you can tell your particles are growing just by watching that peak slide to the right. But it doesn't stop at size, it also brilliantly gives away the shape. If you have perfectly spherical particles, you're going to see a single, clean SPR peak, because those electrons are oscillating uniformly in all directions. But what if you grew nanorods instead? Anisotropic shapes like rods are, of course, longer in one direction than the other. Because of this, they interact with light in two distinct ways, giving you dual peaks, a smaller transverse peak for the short width of the rod, and a larger longitudinal peak for the length of the rod. Just by counting the peaks on a screen, you know if you've made spheres or rods. How cool is that? Application 3 is all about checking stability. In a perfect scenario, your nanoparticles stay perfectly separated and stable in solution, which gives you that sharp, normal SPR peak. But if things start failing and they clump together, a total nightmare called aggregation, their individual electron clouds start messing with each other. This drastically alters their optical signature. The peak widens out, flattens down in a process we call damping, and it exhibits a massive red shift. Visually, your liquid might suddenly shift from red to blue or purple. This is an instant red flag for a researcher that their particles are clumping up and losing stability. Our fourth and final application steps away from noble metals and looks at semiconductor nanomaterials and quantum dots. Now, materials like titanium dioxide or zinc oxide don't rely on SPR. Instead, we use UV vis to find their optical band gap, which is basically the energy required to make them conductive. By finding the absorption edge on the graph, we can use mathematical models called talk plots to calculate this exact energy in electron volts. And when it comes to quantum dots, UV vis perfectly captures quantum confinement. As the physical size of the quantum dot shrinks, its absorption edge shifts drastically. This shift actually causes the liquid to change colors right before your eyes, starting at deep red, shifting through yellow, and finally hitting bright green and blue as the particles get smaller and smaller. Section 5. The ultimate nanotech tool. So, basically, the language of these peaks translates directly into hard physical data about your nanoparticles. Lambda max, the peak position, that tells you the size and shape. The height, or peak intensity, that's your concentration. The peak width, often referred to as the full width at half maximum, tells you if the particles are nicely uniform or if they're starting to aggregate. And finally, the absorption edge hands you the optical band gap for semiconductors. It's literally like the data curve is speaking directly to you. When you stack up all these advantages, it is absolutely clear why UV vis spectroscopy is a complete game changer in the lab. It is fast, it is incredibly simple to operate. It is non-destructive, so you aren't ruining your precious batch of nanoparticles just to measure them. It only requires a tiny drop of sample. It allows for real-time monitoring of live chemical reactions. And above all, it's highly sensitive to even the most minute changes in a nanoparticle's physical state. To connect the final dots here, UV vis gives us an absorption spectrum. From that spectrum, we pinpoint the lambda max or the absorption edge. And boom, by reading those precise points, you unlock a complete physical biography of that nanoparticle. Its synthesis progress, its exact size, its shape, its stability, and its electronic bandgap. We have taken something completely invisible to the human eye and made it entirely readable. Which leaves us with a pretty inspiring thought to wrap up this explainer. If just shining a few different wavelengths of light through a tiny clear tube can reveal the birth, growth, and structural collapse of things literally 1,000 times smaller than a human hair, what other invisible mysteries is spectroscopy waiting to uncover? Thanks for joining me for this explainer. Keep asking questions and keep exploring the invisible.